Welcome, everyone. My name is Jessica Blaustein. I'm the Senior Director of the Healthcare Partnerships team at the Muscular Dystrophy Association. Thank you for joining us today for this MDA Industry Update webinar and for all that you do for MDA and the neuromuscular community. This webinar, titled LGMD2I R9 Disease State Overview, Biomarkers, and BBP 418 Clinical Update, is sponsored by BioSolutions, a BridgeBio company. MDA's Industry Update webinars are programs which provide an opportunity for the medical community to receive information and updates impacting care and treatment directly from pharmaceutical and biotechnology companies. These webinars are just a part of MDA's medical education resources. We encourage you to visit the medical education pages of MDA.org. You'll find resources including MDA's clinical research awareness programs, Grand Round webinars, our MDA monthly report e-newsletter, and much more. Now, I'd like to introduce our presenters for today's webinar. We'll hear first from Dr. Carla Zingariello, a pediatric neurologist at the University of Florida, and then from Dr. Douglas Sproule, the Chief Medical Officer at ML-Bio. They'll share information about LGMD2I R9 and BBP 418. Now, without further ado, I'd like to turn it over to Dr. Zingariello.
Next slide. Great, thank you so much. So we're going to talk today, you might want to go back a slide there. We're going to talk today about the limb-girdle muscular dystrophy disease, give you an overview of the disease and patient experience. Next slide. So muscular dystrophy, as many of you know, is quite diverse, and there are several major types, including myotonic dystrophy, Becker and Duchenne muscular dystrophies, congenital muscular dystrophy, facioscapulohumeral muscular dystrophy, or FSHD, and limb-girdle muscular dystrophy, or LGMD, which we'll be focusing on today. So limb-girdle muscular dystrophy primarily affects proximal skeletal muscle, and this results in progressive muscle weakness. It includes multiple genetic subtypes, which have differences in their age of onset, symptoms, comorbidities, rate of progression of disease, and treatment. And in addition to impacting skeletal muscle, can also lead to cardiomyopathy and respiratory issues. Next slide, please. So the limb-girdle muscular dystrophies are genetically distinct diseases with some clinical overlap. The old nomenclature utilized a 1 to indicate autosomal dominant inheritance and a 2 to indicate autosomal recessive inheritance, followed by a letter for the specific disease type. As you can see, the vast majority of limb-girdle muscular dystrophies are autosomal recessive and eventually ran out of letters in the alphabet, leading to 2017 European Neuromuscular Center Consortium workshop, which introduced updated nomenclature to classify the limb-girdle muscular dystrophies. And each subtype is driven by a variant in a particular gene. So, for example, LGMD2I is also known as now LGMD2R9 or LGMD2R9 FKRP related, where FKRP is the causative gene. Next slide, please. So we're going to focus specifically on LGMD subtype 2I or R9. We'll talk a little bit about the molecular pathogenesis. So in healthy muscle cells, we have glycosylated alpha-dystroglycan that bridges the muscle cell membrane, you can see on the bottom of the first figure there, to the extracellular matrix. So FKRP, or fukutin-related protein, is present to some degree in patients with LGMD2I or R9, and they do retain some function. However, in these affected muscle cells, hypoglycosylation of the alpha-dystroglycan results in loss of this alpha-dystroglycan laminate binding. And so you have shortened and dysfunctional glycan chains that break down, leading to injury to the muscle cell and subsequently a muscular dystrophy. Okay. We can focus on a little bit more, or we can move to the next slide. So for LGMD subtype 2I or R9, even within this subtype, there's genetic diversity and diversity in the clinical presentation. So about 2/3 of patients with LGMD 2I or R9 are homozygous for what's called the founder variant or the L276I genotype. And this results in a slightly milder phenotype with a later onset, typically in late childhood or adolescence. And they present with lower limb proximal weakness, may or may not have calf hypertrophy, may have some muscle pain, and often have an elevated serum CK. There is loss of ambulation in a quarter of patients by age 40. And then non-invasive assistance is required by about 10% by age 40, and invasive assistance required by less than 1%. Cardiac dysfunction is present in about 30% of these individuals. In the other FKRP genotypes, so these include both one copy of the founder variant and another variant or two copies of another variant. These patients have an earlier onset of symptoms as young as 4 years old with lower limb proximal muscle weakness. They can have calf hypertrophy, muscle pain, and often have an elevated CK. Their symptoms progress throughout childhood, leading to loss of ambulation in most of these patients by age 20. Additionally, they have respiratory decline with invasive assistance required by about 5% by age 30, and cardiac dysfunction at higher frequency in about 60%. Next slide, please. So I want to talk a little bit about the patient experience, both with leading to diagnosis and challenges in getting to a neuromuscular specialist, or delays in recognizing symptoms, and then the burden of disease. So for those with the earlier onset, the children, parents may notice some mobility problems, clumsiness, difficulty running or keeping up with their peers. In adults, they may notice they have difficulty with standing, rising from a chair, with endurance in walking or running, and realize essentially something is wrong and will reach out to friends or family or go to the internet, and leading initially to a visit to their primary care provider, a referral either to a neurologist or perhaps an orthopedic specialist. And there may be a delay at this point, in terms of getting to the right provider. But ultimately, particularly now in the era of easy access to next-gen sequencing gene panels, will ultimately lead to a diagnosis of LGMD2I or R9. This, of course, can lead to a patient feeling relief for identifying the cause of their symptoms, but once they recognize there is no disease-modifying therapy, can lead to some anxiety and sadness. There are support organizations such as MDA or Speak Foundation, amongst others, and certainly clinicians can help these patients access resources which are very key, including physical therapy, occupational therapy, other medical specialists, and aid with everyday accommodations for home, work, and school, and needed equipment. And also important is exploration of clinical trials, which we'll be talking about, and health promotion, exercise, diet, and stress management. Next slide, please. So current estimates suggest the prevalence of LGMD2I R9 subtype is about 7,000 between the United States and Europe. And diagnosis, as we mentioned, is confirmed through genetic testing, and this is available through a number of testing programs for muscular dystrophies, as you can see listed here, amongst others. Next slide, please. So I want to touch upon standard of care for LGMD2I, which is aimed at symptom management, and this includes physical therapy to prevent contractures and maintain mobility, cardiac monitoring and medical management of cardiomyopathy, respiratory monitoring and respiratory support, pain management, and low-impact aerobic exercise, as well as touch upon some of the unmet needs, as there's no approved disease-modifying therapies currently, as well as a lack of well-defined standard of care, including management of cardiac and pulmonary symptoms. There are no approved therapies for any of the limb-girdle muscular dystrophies currently. Next slide, please. Okay.
Dr. Zingariello, thank you so much. This is Doug Sproule. I'm the Chief Medical Officer at BioSolutions, and I wanted to take this opportunity to kind of build upon what Carla just spoke about and talk a little bit about our clinical program and how we hope to help continue contribute to addressing that unmet need in the LGMD2I R9 community. So we can go to the next slide. I want to talk about our product under development. And again, we're going to talk about BBP 418. It's a product that has not been approved by any regulatory authority in any country. I'm going to talk about some of the preclinical data and obviously take it for what it's worth. BBP 418 is being developed as a substrate therapy that's designed to treat LGMD2I R9. And I'll talk a little bit more detail about the specific mechanism of action of this product. It's provided in the form of granules that are dissolved in water and consumed twice daily, so typically in the morning and the evening. This product is under active study. There have been approximately 100 patients and other individuals have been exposed to this product, and it's been overall well tolerated to date. There have been there have been no significant dose-limiting toxicities. There have been a few discontinuations in our phase 3 study, but otherwise no serious adverse events that have been related to BBP 418. I'll talk a little bit about that as well. We have an ongoing phase 2 study, which is an open label study. I'll talk a bit more about that, as well as an ongoing double-blind randomized controlled phase 3 study. We've seen from the existing data to date, we've had very encouraging biomarker trends and positive trends in functional outcomes that I'll talk about that make us very excited to see how this product will perform in a hopefully label-enabling double-blind randomized controlled phase 3 study. If you can go to the next slide. I want to build upon a little bit of what Dr. Zingariello talked about with regard to the underlying disease and disease mechanism and how our product addresses this unmet need. BBP 418 is being investigated as an upstream substrate supplementation, which is meant to drive increased residual activity of the mutant FKRP enzyme. And on the left side, you see what happens in obviously a simplified fashion in the disease state. So what happens is that you need to have functional FKRP enzyme activity to effectively glycosylate alpha-dystroglycan. This is a critical glycoprotein to stabilize muscle cells binding to extracellular ligands, as was discussed. In the context of a biallelic mutation to FKRP, which results in a partial loss of function, this leads to many of the alpha-dystroglycan species being under or hypoglycosylated, and this results in ineffective function, which increases muscle cell susceptibility to damage and ultimately leads to the classical features of a muscular dystrophy that we see in this disease. And so you can see in this cartoon that you have normal physiologic levels of CDP ribitol in the cell, which is the substrate of FKRP, but because of that mutation, it's ineffective in fully glycosylating alpha-dystroglycan. Our therapeutic approach is to supply supraphysiologic levels of a synthesized pharmaceutical-grade form of ribitol. This is in turn converted by an enzyme called ISPD into CDP ribitol. And while the FKRP enzyme is mutated and dysfunctional, some residual function is obligatory for fetal life and fetal viability. And so you can, if sufficient, you can harness that residual enzyme activity with increased substrate to drive forward in a Michaelis-Menten fashion, increased glycosylation of alpha-dystroglycan. And if this was sufficient, it could potentially slow, halt, or allow recovery from the underlying clinical disease. And so this is what I would like to call a simple solution to an elegant, or simple and elegant solution to a complex problem. So if you can go to the next slide. And we have an extensive clinical program that I can't go through in detail in the interest of time, but we've conducted a large natural history study in collaboration with the GRASP consortium. We've conducted 2 phase 1 studies, a single and multiple ascending dose study, as well as a food effect study in healthy volunteer subjects to establish the basic safety and pharmacokinetics of this product. We have an ongoing long-term open-label phase 2 study that I'll talk about in some detail. And we have a large ongoing 36-month double-blind placebo-controlled study that is ongoing in the United States, Europe, UK, and Australia, and is at this point fully enrolled, and the patients are proceeding through the study as we speak. So if you can go on to the next slide. One of the things that's something that's been well described from the natural history study that we conducted in collaboration with the GRASP consortium is the relevance of measuring glycosylated alpha-dystroglycan levels as a marker of disease severity. And so what this figure shows is the patients from the natural history study. Again, this is open label patient, a natural history study, non-therapeutic study. And what we see is that on the left are patients who are homozygous for L276I mutation. These are the relatively milder form of the disease. And these patients have a average glycosylated alpha-dystroglycan level of approximately 10% of a healthy control reference standard. And in comparison, patients with other genotypes, again, we know from the natural history of these patients tend to have a much more severe presentation. And commensurate with that, we see that patients with other FKRP genotypes have approximately 5% of that reference standard, glycosylated alpha-dystroglycan levels. And so there's a significant reduction in glycosylated alpha-dystroglycan that's seen in patients with LGMD2I R9, as we would expect. And the severity relates and tracks with the known population severity of the disease. So if you can go to the next slide. So then the question is, what happens in the context of BBP4 and 8? And this is data from our phase 2 study that we presented previously. On the left side, you see the change in glycosylated alpha-dystroglycan levels following administration of BBP4 and 8. On the left side of the figure is the patients who are homozygous for L276I, and the patients in the phase 2 study changed from an approximately 16% of that reference standard at baseline to approximately 40% that was confirmed on repeated sampling over up to 15 months of longitudinal follow-up. Similarly, patients with other FKRP genotypes, they started from a lower baseline, which is what we would expect, approximately 6%, and they increased to a median of around 10% following administration of BBP418. So we're able to see and demonstrate that this therapeutic strategy has an impact on increasing glycosylated alpha-dystroglycan levels across the genotypic spectrum of this disease. We are also seeing an impact on other markers of muscle breakdown and injury. Creatine kinase is a widely used and well-known marker of muscle breakdown, obviously a non-specific marker, but one that would be expected to improve in the context of a therapy that improved the stability of the muscle membrane. And we see this with a significant drop that is sustained on repeated sampling over time. We see a more than 80% reduction in creatine kinase from baseline in patients treated over the longer term with BBP418. So this obviously is very exciting early data that suggests that there's an impact on the level of cell that then translates to the level of the organ itself. If you can go to the next slide. Obviously, we don't really care about creatine kinase levels or glycosylated alpha dystroglycan levels. We do care about how this has an impact on the clinical status of patients. We are seeing a signal, albeit obviously it's a small study, it's open label, there's all the biases that need to be recognized, but we're seeing an improvement in performance on ambulatory measures such as the 10-meter walk test, the 100-meter time test, as well as motor function measures such as the North Star Assessment for Limb-Girdle Muscular Dystrophies, or the NSAD, when compared with the trajectory that we saw in the GRASP Consortium led natural history study, the MLB 01001 study. And so this, again, suggests that there is the potential of this therapeutic strategy could stabilize or potentially allow prospect of improvement in patients treated with this therapeutic strategy. So if you can go to the next slide. So this, gave us the confidence and the proof of concept to commit to a large double-blind randomized control trial that is called Fortify. This study is now fully enrolled. It enrolled 112 individuals across the United States, the United Kingdom, Australia, and Europe. This is, as mentioned, a double-blind randomized control trial, 2 to 1. So 2 patients receive drug to every 1 patient that receives placebo. And these patients are followed for 36 months. It's a long and heavy commitment that we're asking of patients in this study to allow us to very comprehensively and conclusively demonstrate the impact of this therapeutic strategy. We expect that patients will be offered the opportunity to enroll in a separate open label extension study, which will allow and provide ongoing access to BBP 418 for patients who participate and complete the phase 3 study. At 36 months, our key primary endpoint is a North Star Assessment for Limb-Girdle Muscular Dystrophies. We're also going to be looking at ambulatory measures, measures of pulmonary function, upper limb function, as well as the 2 biomarkers I mentioned, glycosylated alpha dystroglycan levels in particular, as well as serum CK. If you go to the next slide, I think one of the things that's obviously a major question when you have a 36-month study is how can we, potentially create an avenue of access for patients in a sooner and more effective fashion. And so we are planning to conduct an interim analysis at the 12-month time point when at least 42 patients have completed at least 12 months of study. We expect that we will execute this interim analysis later this year. We're going to be looking specifically at biomarkers, the change in glycosylated alpha dystroglycan levels. We'll also be looking at change in creatine kinase and selected clinical endpoints in a formal statistical hierarchy that will allow us to have an effective conversation with the regulatory authorities about pathways to an accelerated approval process. So obviously this is something we're very interested in and excited about. We'll have to see how that data emerges as it's available. So if you can go to the next slide. And this is kind of the linchpin of how we're thinking about things. As many are aware, there's been a major shift, and we hope it continues in the approach that regulatory authorities are taking towards rare diseases, particularly in the neuromuscular space. And we are intending and planning to perform an interim analysis that will include measurement of glycosylated alpha dystroglycan. And we are working avidly with the authorities at this point to develop and support its use as a potential surrogate endpoint of clinical impact. And so we believe that this approach does have potential to allow us to pursue an accelerated approval pathway based on the surrogate endpoint of glycosylated alpha dystroglycan levels and the change in response to a therapy in LGMD2I R9. So certainly something that we are very interested to see. And we've received positive feedback preliminarily from the FDA on their willingness to engage us in this manner. And obviously there'll be much more to say once the data emerges and we've had the opportunity to really dig in with our partners on the regulatory front. So we can go to the next slide. ML Bio is a small company. We are a small operation. We're very lean. But But we're very proud of what we've accomplished, and we're excited about the path ahead. We've done an amazing amount in what we consider a short period of time. I know the community considers it glacial, but we've partnered with our academic partners to complete a natural history study in LGMD2I R9 across the U.S. and Europe. We've completed 2 phase 1 trials, a phase 2 study that's ongoing, continuing to contribute data. We've achieved and been granted fast track designation, rare pediatric designation by the FDA, and orphan drug designation by both the FDA and the European Medicines Agency. And this has allowed us to commit and perform a large phase 3 study that is now fully enrolled. And so we're extremely excited about what we've done, and we're extremely excited about the path ahead. And at this point, we expect an interim analysis to be conducted at some point in hopefully later this year. Our last patient last visit will occur in 2027. And we are eager and will continue to engage with healthcare practitioners and patient advocates in the patient community to continue to learn what we can do better, what the needs are of the community, and how we can be as good a partner with the neuromuscular and LGMD2I R9 community as we move forward. So I want to thank everybody for allowing us to speak. I think we have some time for Q&A if there's anything.
Yeah, we have 3 questions in the chat that I can get to you. So the first question is, if you can comment, are there any plans for BridgeBio to address LGMD2A RI? And if yes, what's the timetable?
So the quick answer to that question is no. We have a single product at this moment, and that is the development of BBP-418, which is a synthetic version of ribitol. This is a therapeutic approach that's appropriate for patients who have mutations of FKRP, which is LGMD2I R9, and also potentially FKTN, which is and ISPD, which are 2M and 2U. Unfortunately, this therapeutic strategy would not be a relevant approach for other forms of limb-girdle muscular dystrophy. That said, there is relevance for patients with other diseases. As you see how the pathway moves forward, the approach that regulators have towards these approvals, the ability to utilize natural histories. These are all valuable things that have cross utility to other companies and to other programs as therapies are developed, to have a better understanding of how we can most rapidly and accelerate the development. And so it has, while it's not necessarily applicable to an individual patient or with another form of limb-girdle muscular dystrophy, has broad impact across the whole community.
Thank you. Not a question, but a comment. Another person says, not a question, but thank you for including the poll in your assessments. It will help with insurance authorization in the future.
Yeah, one of our goals is to try to create as broad a data, a broadest study that will allow the broadest possible access for patients. Our goal is not to have this therapy limited to a certain subset of the relevant patient population. We hope that'll help.
I think that's all the questions that we have time for.
Great. Thank you so very much to Dr. Zingariello, Dr. Sproule, Anna, Sibel, the entire ML Bio team. Thank you for supporting this program and bringing this information to our medical professionals. Thank you also to everyone who took time out of your day to attend the presentation today, and we hope you'll join us for future industry update webinars. Have a great day. Thank you.